being administered today, and I think in a short while, Honorable PM is going to launch the vaccines. So that's a very positive development in the country, and we are also seeing vaccination in other countries as well. But there is also a trend of concern which has come up, not so much in India, but globally, with respect to new variants of SARS-CoV-2 being reported. Now, what are these variants and what are the implications? And should India be concerned or not be concerned? So these are some of the questions on top of everyone's mind. And we at CSIR, as usual, have been engaged in COVID-19 research. And also this is the problem also we are addressing. And we also have industry partners who will talk about how industry is coming up with solutions towards addressing these challenges during COVID-19. So today we have a galaxy of speakers. So we have our DG CSIR, Dr. Shekhar C. Mande, and Dr. Tej Sopati from CCMB, Dr. Anurag Agarwal from IGIB, and Ms. Avani Singh from Spice Health. So before we go on to the main talks, I request Dr. Shekhar Mande, DG CSIR, who's been very active in communicating on the various COVID-19 safety measures, and you must have seen him on TV many times coming and talking about the science. He never says no to any communication on COVID-19. He's been the driving force behind these webinars. So Dr. Mande, please give us an overview of what do you think on this new variants and where we are going.
Thank you, Geeta. First of all, welcome all the listeners on behalf of CSIR in this webinar. As Geeta told you correctly, that CSIR has been attempting to bring a lot of information about COVID-19 through these webinars. Time and again we have organized, and many people have joined us in this particular series. And we are essentially trying to inform the public about COVID-19 and what are its dangers and what are the possible solutions, how we should behave, and so on and so forth. So I welcome my two of my colleagues, senior colleague Dr. Anurag Agarwal and younger colleague Tej. Tej, I've seen him from his very young days as a student. And I also welcome a very distinguished guest, Avani Singh, amongst all of us. We all know that humanity was faced with a crisis exactly about a year ago. In fact, 7th January 2020, the first of the sequences was made. And on that day, it was realized that the crisis that we are facing is because of severe acute respiratory syndrome, coronavirus, what was called 2, because the CoV-1 was already there in 2003-2004 in the parts of East Asia. In the era that has actually followed since then, since 7th January 2020, because of the 1 year severe acute respiratory syndrome, coronavirus described as a frantic year, was called 2, or because the CoV-1 all the scientists and the healthcare professionals around the world have literally in the parts of day and night East Asia to find what are the potential solutions. In the era that has actually followed since then, since 7th January, we actually worked day and night to bring 2020 because of the 1 year severe acute respiratory syndrome, coronavirus described last 100 years as a frantic year, was called 2, or because the CoV-1 and all the scientists and the healthcare professionals around world have literally the parts of night East Asia to find what are the potential solutions. In the era that has actually followed since then, since 7th January, actually evolution of the virus and sequences and evolution of new strains and all. And just to give a very brief 2-minute overview, we all realize that when any pathogen invades a host, both the pathogen and the host try to adapt to each other. The pathogen is trying to establish an infection in the host, and the host is trying to get rid of the pathogen. And what ensues is some sort of a tug of war between the two of them. And the tug of war, they try to adapt to each other through what we call as genetic mutations. So both host and the pathogen try to actually evolve, and what ensues is some sort of a tug of war between and try to see how they can adapt to each other. And the tug of war here is some sort of a try to adapt to each other through what we call as genetic mutations, in which the replication cycle of the cells is much slower. Try to actually evolve, and what implicates is some sort of a tug of war and try to see how they can adapt to each other and to accumulate mutations. And we are therefore naturally at a disadvantage as a human host, that by the time we adapt to the virus is going to be thousands or probably millions of years. And we do indeed have examples in the human genome. We do have actually some traces of old viruses. In the human genome sequence, very fascinatingly, has traces of some of the very old viruses million years ago that have integrated into the human genome. But it takes that kind of a time. Well, the virus replicates every day, and therefore it has a chance of replicating and accumulating mutations every day. And therefore, it is probably evolutionary more advantageous for it to mutate and adapt to the human host. But then we, of course, have a beautiful immune system that takes care of the virus. And our immune system is at the virus all the time. And our immune system is extraordinarily capable of getting rid of the pathogens such as this. And therefore, the interplay that actually happens between the host and the pathogen is the most fascinating aspect of science that what we see. And the mutations that we are seeing in the virus is a direct consequence of this interplay between the host and the pathogen. And we don't need to be worried about it. Our immune system is perfectly capable of dealing with it. Only thing is we have to make sure is that we should enable our immune system, or we should boost our immune system for it to be able to deal with the pathogen. And that's exactly what vaccines, or God forbidden you get infection, drugs, they exactly do help us in doing that particular thing. So there is no need to worry. It's a fascinating scientific aspect. But what we'll try to do is today is bring these fascinating aspects to all of you. And we are very pleased that we have very, very distinguished people. One of the best known faces in the country is Dr. Anurag Agarwal. He is with us to talk about all the mutations and genome sequences and all. I don't think there can be any better authority than him in the country to tell you about genome sequences. We also have very bright and young Divya Tej with us, who will tell us about this thing. And Tej has been directly involved in the work on COVID-19. And of course, we have Avni, to whom we cannot thank enough for coming forward and partnering with us in bringing these very valuable solutions to the public. So we have a very exciting time in the next 1.5 hours or so. So look forward to these discussions. Thank you all very much.
Thank you, Dr. Mande, for that very nice introduction and laying the context for this webinar. And now we move to Tej. Tej, as Dr. Mande described, is a scientist at CSIR CCMB, and he's been very much involved in the COVID-19 from the beginning. And over to you, Tej, please.
Yeah. My slides are visible?
Yeah, they are visible.
Okay. Good morning to all of you. I want to thank both Dr. Mande and Dr. Raisam for first hosting this webinar and also asking me to be a part of it. So in the next few minutes, I'm going to talk to you or give you some introduction about how we go about analyzing these viral genomes. How do we sequence and analyze them to understand these viral genomes better? Okay, my slides are not moving. Okay. So yeah, I don't think this introduction needs to be given at all. But what was striking to me was that so I had used the same slide about a month ago, and that time this number on the left read 63 million, and now to look at it pop up to 93 million was really striking. And like just in about 40 days, we have 30 more million cases, and I think it's a reminder that all of us should take due diligence and responsibility to contain the spread, because as Dr. Mande was mentioning, the virus is capable of mutating much faster than we can. So we should not give it enough chance to do that. Or as the more we minimize that kind of a playground for the virus, the better it is for all of us. But yeah, given the extent or the global nature of COVID-19, it is not exaggerating to call this the pandemic of the century. And at CSIR and other labs in the country, we have been trying to tackle this pandemic that's been put on us in a variety of ways. For example, we use viral genome sequencing to understand how the sequence of the virus itself affects its biology, as well as its infectivity or spread. And also we use its sequence to track and surveil where all the virus is spreading and so on. And also to see if these kind of mutations that occur in the genome have any effect on therapeutics such as vaccines or drugs that may be developed against it. In addition to that, we also look at the host genome itself. As the DGCSR said, it's a complex interplay between the host genome and viral genome, and a tug of war on who can adapt to what. So there may be already host genetic factors that could be having a role in disease susceptibility or how the disease will progress further. In addition to that, we also look at the gene expression profile of the host itself to see, to understand how the body immediately responds to this viral infection and whether that has a role in the disease progression. And finally, we're also tackling something called the co-infections, which means that if the host already has other pathogenic infections, whether that will cause more complications or not for COVID-19. But today I'm going to mostly focus on the first aspect of it, in which CSR has been active from the very beginning. And I'll take you through some slides to help you understand how we go about it and what it means. So as we all now know, COVID-19 is caused by this RNA virus called SARS-CoV-2, which is a single-stranded RNA virus, and it's about 30 kilobases long. So the RNA is made up of ATG or AUGC, and it has about 30,000 of them organized in one linear fashion. And two proteins called the ORF1A and 1B, which occupy most of it, they code for most of the proteins that are important for the viral packaging and so on. And then you have various structural proteins. For example, you have the spike protein, which is what is the top of or the outermost part of the virus, and it gives it the crown-like appearance of the virus. And the spike protein interacts with the ACE2 receptor of human cells, and that is what is important for the successful entry of virus into the host. So, and this 30 kb, 30 kilobases, when I say kb, 30 kb genome is what we are interested in to understand how that is mutating. So before I go about it, I'll briefly explain some terminology I'll be using in this talk. So first is a mutation. Mutation is nothing but a change in this genetic sequence. As I said, there are 30,000 bases of it. One of them can change into something else, or it can end up getting deleted, or maybe an extra base can get added. And these are a natural process of any organism. And because microorganisms replicate much more or much faster than organisms like us, they also show many more mutations than something like humans. And most of these times, these mutations do not have any consequences, or sometimes they're actually, or many times they're bad for the virus. But occasionally, they give an advantage for the virus. In a sense that maybe it will enable the virus to, infect more or spread more or stuff like that. And whenever that kind of an advantage is seen, slowly you will see that virus carrying that particular mutation overtakes others because it has a fitness advantage. And a strain is nothing but a genetically distinct type of virus, which means that this particular, so when I call a viral strain, what it means is that it has one or more mutations in its genome. And we also often call it a variant, and it is used in the terms similar to that of strain, but sometimes we also could use the term variant to talk about a single mutation. And finally, we talk about clades, which are nothing but a group of viruses that share a common ancestor. So what it means is that you have, let's say, a mutation 1 happening in the beginning, and two viruses that come from the both carrying mutation 1. One can acquire mutation 2 and the other can acquire mutation 3. But if you see, both of them have a common mutation 1, and most likely they have come from the common ancestor which carried that mutation. So a group of viruses that are coming from a common ancestor is known as a clade. And all this comparison, when I say mutation or a difference or something, for SARS-CoV-2, we basically use the Wuhan strain, which was sequenced, which was first released on 7 Jan 2020, sampled from December 2019. That is what is used as the background for comparison. So any change that we see with respect to that strain, that is what we call a mutation currently. So in the next few minutes, I'll briefly explain how we actually decipher the sequence of SARS-CoV-2. There are a few approaches. The first one, which is what we all started with in the beginning, is called the direct RNA sequencing. So what essentially happens is once you have a positive sample, which is a swab sample of a patient, we extract RNA from the entire swab, and then we directly go for something called next generation sequencing. And that's it. So there are no other steps. So it's very simple and straightforward. And because we are not doing any kind of manipulation to the sample, we have no bias in the kind of details we get from the virus. And because the swab RNA contains viral RNA as well as the human RNA, in the same shot, not only do we understand the sequence of the virus, but we can also understand what genes are expressed in the host and so on. But there are some limitations to this. For example, because it has both viral and host RNA, we have to sequence it much more than a method which sequences just the viral RNA, which makes it more expensive. And the success actually depends on how much viral RNA is present in the sample. So if a sample had very tiny amount of viral RNA, of course, most of the data we get will be of the host and not of the virus. So then we cannot understand the sequence properly. So its success depends on how much virus is present in the sample. So that's the reason this is not used as much. The other method basically uses a way to enrich the viral RNA in the sample. So what this does is, because of how the hybridization between RNA or DNA molecules work, we can use, because we know the sequence of the virus, we can use something that is like the complementary of this particular virus, and we can use that to fetch out just the viral RNA from the samples. So we have the total swab RNA, then we add this complementary mixture, which will enrich the viral RNA, and all that is then collected and sequenced. So because of this enrichment step, of course, the success is often better than direct RNA sequencing. But then there is this extra process of having to do this enrichment, which takes time. And it's also slightly more, although cheaper than direct RNA sequencing, it is still expensive. But it also gives this other advantage that this complementary thing need not be just against SARS-CoV-2, but it can be against other pathogens. So let's say someone comes with some respiratory symptoms, and you're not sure whether they have SARS-CoV-2 or some other respiratory virus. You can use a panel which targets all respiratory viruses, and in one go you can identify whether they have SARS-CoV-2 or anything else as well. So this is the other approach. And finally, we're going to talk about the approach that is the most commonly used. So this is nothing but the amplification of viral RNA. So the first step, the first method I talked about was no enrichment, nothing, direct sequencing. The second is where you are enriching for your target. And third method is where you're not enriching, but you're amplifying your target, which means that you're increasing the number of copies of your viral RNA in the sample so that you can sequence more of it. And that is done using a technique called PCR, which uses something called primers, which bind to this viral RNA and generate multiple copies of it. And what we do in this particular method is that we use primers which target 400 base pair fragments of the entire 30 kb of the virus, and we amplify all of them, and then we sequence them. So this PCR-based approach was originally standard by a network called Arctic Network in the UK, and now we have other commercially available alternatives such as QIAseq or COVID-seq. All these are the same approach. Essentially, they use a PCR-based method to amplify the viral genome so that you have many more copies of it, and then we put it through sequencing. So this is because of how much it is amplified, you only have to sequence very less. So that's why it's cheap, and you can do thousands of samples in one go, and there's no complex workflow. But of course, because there is this amplification that is happening in multiple steps, there is something called a PCR bias that can often introduce some bias, but for the most part, it is okay. And as I said, this is the most commonly used approach. So even if you look at a leading consortium such as the COG consortium in the UK, who have sequenced more than 150,000 samples, they've mostly used this particular approach for the sequencing. So now I'm going to talk briefly about what has been happening last year in India in terms of the variants that we see. So as I mentioned, CSR labs have been actively involved in genome sequencing from the very beginning. In fact, I remember at CCMB, end of March was when we received the first swab samples, and April 6th or 7th, we already had, via direct RNA sequencing, we had the first genomes out. Since then, about 6,500 genomes have been submitted to public domain from India, and more than half of them are submitted just by CSR labs. So, and all these various methods that I talked about, they have been well standardized in our labs, and we use a variety of platforms, the latest platforms such as Illumina or Oxford Nanopore platforms to achieve this. And we also have set up a streamlined data analysis so that we don't just generate the data, but we immediately see what is going on and what it means in terms of either circulating strains or the new strains that are coming up and so on. So initially at around May, when we were analyzing this data, the picture of the virus distribution was something like this. So in this plot on the x-axis, we have time from about Feb, when the initial samples were sequenced, and up till June. And each row represents one state, and the top row is actually of the entire country. And the color indicates which particular clade the virus belonged to. So as I said, clade means there are a group of viruses which came from a common ancestor. But at that time, I mean, now they're colored green and we're calling it IA3I, but at that time, 40% of these genomes went into so far an unclassified clade. So this was done in collaboration with IGIB and us, and we called it the IA/A3I clade. And so these were actually, this particular clade was an India-specific clade, although its ancestors most likely came from East Asia and other countries. And this particular clade basically is identified by about 4 different mutations in its genome. And on the right side, if you look at this particular tree, the way it is seen, like you have a common central point, and then you have this bursting kind of tree from all over. What this indicates is that this particular clade of the virus entered India and then spread across the country in a fast manner. So and because of the high number or high frequency of the clade observed in India, we believe that most of the initial infections in India happened or of this clade. And one of the mutations that we saw was present in something called RDRP of the virus, which is actually important for the virus to replicate. And we predicted it to be deleterious, which means that, as I said, the mutations can often be disadvantageous for the virus. And we predicted that this was one such mutation. And if our prediction was correct, what we should see is that slowly this virus should disappear because other viruses which do not have this disadvantage will take over. And that is what we see currently happening. So this is a similar plot, but till October, where we have good genomic data. And you can see that although in India, initially it was all, or there was a mix of green and blue, where green is actually the I/A3I clade, and blue is the other clade called A2A clade, which was the globally predominant clade. Slowly, it has become mostly blue dominant, which means that we don't see this particular clade in circulation anymore, and it's mostly the A2A clade that is in circulation. And in fact, 70% of all genomes that we have now sequenced in India fall into this particular clade. And they actually carry this other mutation called D614G in its spike protein, which also made a lot of news last year that it leads to an increased spread or infectivity. That was the scenario till last year, and around that time is when we started seeing this appearance of new clades. So I'll briefly talk about few of them that are now currently circulating. So the first one is called 501Y.V1, which was identified in the UK first, and so it first appeared in September 2020 in various regions of the UK. And then they've seen that it very quickly became the most dominant strain in the UK. And currently they're saying that almost 80% of all new cases have are of this particular clade. And the reason why people started panicking is because it is actually shown to be about 70% more transmissible. And that they think is because of various mutations that are there in its spike protein. I'll cover briefly later on. Although they don't see a specific increase in severity and mortality, they see it has a very quickly spread globally because of international travel. And we already know that we have more than 100 cases identified in India at present, which belong to this particular clade. The other one is called 501Y.V2. So the reason why they are all called 501Y is because they carry this particular mutation N501Y in its spike protein. But the other mutations are different. So this particular clade was identified in South Africa first, and in about October 2020. Although they first saw that it belonged to a lot of samples from the eastern part of South Africa showed this particular set of mutations, they've seen that again this showed a very rapid spread and now is the dominant clade both in eastern and western provinces of South Africa. And there is also some early evidence pointing to the fact that this particular strain can escape neutralizing antibodies. I believe Dr. Anurag will cover these things in more detail, but so that was the reason why this is also of concern. We are yet to see the presence of this strain in India. And the third one, which also carries this N501Y, and it's called V3 version of it, and it was identified in Brazil first, very recently, something like early December 2020. And there they have seen that 65% of all new samples that they've sequenced in December belong to this clade, and they share a lot of mutations with the V2 that I've shown earlier. And it's currently also seen in one other country, Japan, and it is still not detected in India. So what is interesting with these particular set of circulating variants is that this is an enlarged version of its spike protein of all the three variants. And first thing you see is this common N501Y mutation in all the three of them. But other than that, particularly for V2 and V3, so you see a lot of other mutations which are common. For example, this E484K and L18F. And these are mutations that did not come from a common ancestor. So all these clades actually independently evolved and acquired these mutations, which means that this is a beautiful example of convergent evolution, what we call as convergent evolution, which means that something advantageous for a species, they end up independently acquiring those traits, although their common ancestor did not share it. And this is one such example, meaning that this particular set of mutations is somehow giving a good fitness advantage for the virus, and which is why it is seen so often in multiple variants circulating all across the globe. And also, in fact, this particular mutation N501Y has also been shown in some mouse studies where they tried to infect mouse cells with virus, and the virus actually accumulated this mutation in mouse cells directly, again showing that that particular mutation somehow is advantageous for the virus. So this is the global scenario. Very briefly, I'll talk about one particular mutation called N440K, which we have seen in some AP samples. So this work comes from Dr. Vinod and Sridhar's lab at IGIP. So in their particular analysis, they've seen that in Andhra Pradesh, we see this particular mutation appearing as clusters. And when at the height of sequencing, you can see that more than 40% of the samples from Andhra Pradesh actually carried this mutation. Of course, now we don't know the current trend because there are no more new sequences coming from this region, or for that matter, all over India. But this could potentially be another variant which probably has some kind of fitness advantage and could probably become the dominant clade, maybe in Andhra Pradesh, and if we don't contain, maybe elsewhere. What it means in terms of severity or anything, we still don't know. But these are the kind of things genome sequencing will help us identify to see what is going on and whether a given set of mutations will give fitness advantage to the virus. And if it does, how do we contain it? How do we stop it? So all this indicates that we have to continue doing this viral sequencing for some time to come, at least, to monitor these kind of mutations so that we are aware of how the virus is mutating and how we can tackle it. So to that end, a new consortium has now been formed called INSACOG. So this is a nationwide effort to actually continuously sequence these mutations or these viruses so that we can track novel as well as circulating strains of the SARS-CoV-2. And this is actually 10 labs are part of this consortium, out of which 2 are CSIR labs, both CCNB and IGIB. And this consortium aims to sequence 5% of all positive cases in India. So if we have, right now we have about 18,000 cases coming up. So that's about 900 cases per day that will be sequenced. And the idea is to use consistent data generation and analysis pipelines so that this data can be compared across the country easily. And so, yeah, I think that is where I'll stop, and over to you, Geeta.
Yeah, thank you, Tej, for this taking us through the various aspects of the variants, and also explaining the difference between the variants, clades, and mutations, because these many times are interchangeably used, and kind of people who are not in the area would kind of get confused. Thank you for really explaining in detail, and if there are any questions, we'll come back to you later. Thank you. Now we have Dr. Anurag Agarwal, Director IGIP, and as Dr. Mande mentioned, he is a clinician and also a scientist, and he's been leading the efforts on SARS-CoV-2 as part of CSIR and as part of the entire scientific community. And he will take us through and tell us, what are the implications of these variants. So there is certain concern. So should we be worried? When there is also vaccines, would they work on these variants? And what is India doing in this direction? So I request Dr. Anurag Agarwal to take over.
Hi, Geeta. I assume my slides are on the screen?
Yeah, they are, and we can hear you as well. Yeah.
Okay, fantastic. So I'm going to basically, before I get to the point of the strains out there, I won't spend too much time on that. I will take forward what Tej talked about in terms of why are we doing NGS, why we're trying to find the strains, and how should India go about it from a more practical standpoint, which might be relevant for public health. So 2 documents I have used extensively while preparing these slides are from the WHO. One is the interim guidance of SARS-CoV-2 genomic sequencing for public health goals. Please note, it is for public health goals. So everything that I will talk about will be for a purpose of advancing public health. And the second, of course, another guide for implementation for maximum impact. Both are very similar. One is more technical than the other. The starting point is, why do we need to do any of this at all? Recent, and when Avani will speak, she will, I'm certain, talk about the lab we have established at the IGI airport. Why are we making these efforts? Why do we need to sequence or find any strains? Of course, it's important to identify the virus. Sometimes the virus is new, you don't know what it is. Then you want to investigate the spread. Where is it coming from? And can you start understanding how to break the transmission? Third, most relevant for today, study virus evolution, understand changes in its behavior. Now, what are the parts of a infecting virus's behavior that we care about clinically? One is infectivity. How many people with a person who's infected infect further, the R0? That R0 determines the exponent of the rise, which is very high for the SARS-CoV-2 virus. But just to put this in context, while the SARS-CoV-2 virus has very high infectivity, it is nothing compared to measles. So measles R0 crosses 10, while SARS-CoV-2 is still in the range below 3, even the UK variant probably. And the third, which is very important, is immune escape. One of the things that has been good about the virus so far is that people who have been infected seem to have about at least an 80% protection from being infected again. And given that severity of the disease, which is the other reason for us to want to know about the virus, is the disease becoming more severe, is such that for an average person, the risk of a severe outcome after an infection is below 0.5%, and the risk of death is below 0.15%, which is substantially more than the flu, but is much less than, say, Ebola or MERS or even SARS, the original SARS infection. The last, which is important, is to track jumps between animals and humans. It goes in both directions. You can infect an animal and the animal can infect you. And these kinds of jumps often lead to more severe mutations then happen within the same host. So humans will tend to influence the virus similarly, but as it jumps between humans and minks, humans and cats, humans and dogs, there'll be far more mutations. It'll accelerate the entire process. Dr. Mande had talked about that. So if it is so important, why is it not used already? That's the question everybody probably has in their mind, that suddenly it has become very important, but so far nobody was talking about it. 3 reasons. First is cost. For most well-proven systems like the Illumina that you hear about often, it's a very expensive machine. Number 2, it's an obvious outcome of the cost, access. If there are very few places with it, then sequencing is difficult. So most people have no access to such facilities. And the last, which I will be bringing up more, is utility. So far, historically, there have not been enough proven use case scenarios for clinicians. Now, is it because they were not there, or is it because cost and access limited it? I would argue to all of you, the real problem has been that the scenarios have not been formed because the cost and access have always historically been high. Otherwise, many tests that you're already using, for example, tuberculosis, and you're looking at drug-resistant tuberculosis, you're doing PCRs for it. At the end of the day, I cannot imagine a future 5 years down in which we don't routinely use sequencing. We not only identify the pathogen, we identify its drug resistance profiles, we identify where it may have come from, and we do all of that as NGS-based diagnostics, not only for strain identification. And this is going to become progressively more important as we lower the cost and increase the access. So to me, the biggest issue is how to do cost lowering and access improvement. And that's why I'm so happy that Avni will be following what I say. So this is a figure I've taken from the WHO guidance. How do you design a sequencing program? Obviously, when I speak, I'll be speaking a little bit for India. There are 3 types of capacities you need to consider. Do you have a diagnostic capacity? If you don't have diagnostic capacity, either you're planning to use sequencing for diagnostics, that's a bit unlikely if you don't have any diagnostic capacity. And if you have it, you can move to the next step of actually sequencing it. Next, do you have sequencing capacity? And even if you could sequence, do you have the ability to analyze the data? And this is actually quite important. There is no point sequencing and generating mutations if you do not know what you will do with those mutations. If you have no clinical data to tag to those mutations, you have no understanding of what is likely clinical impact, then sequencing alone will be meaningless. And similarly, even if you had clinical data, if you had a sequencing variant, you had no ability to understand the impact on the protein structure, to understand which mutations are meaningful and which mutations are meaningless. It would again not take you very far. And the reason is this. Now, this is a very busy slide. I don't actually expect you to read any of it, but I'll just point out a few things. There are multiple clades, and that was explained to you by Tej. I've put all the numbers S, L, V, G, G, GR, GV. One of the very important ones is a mutation called D614G. It's already taken care of in these clade formation. D614G was the first example of a mutation that increased the infectivity. On top of it, many mutations have taken place. So this green thing is the human ACE receptor. This overall gray thing is the spike protein. Every colored dot that you see, including gray dots that you cannot see, are basically mutations of possible significance on this backbone. Now, among these, only the red ones are the only ones of proven significance. So you can see there are lots of variants. You can see amongst lots of variants, there are a few variants red in color that get into the domain that might be interacting and might be important. So important variants are far fewer than unimportant variants. It is almost impossible to give you definitive examples of which are the important variants except for D614G and what we talk about the N501Y, which come from the UK. Of course, the UK has much more than that. It also has deletions. But again, today is not a good day to talk about that. But let's move on to the real question. There are lots of variations. There are some variations of significance. Do we need to worry about it? Now, there are three types of reasons to worry, right? Infectivity, severity, and immune escape. Now, in terms of infectivity, the same things that you would do for any respiratory virus will continue to work even if infectivity rises, except that you need to do it more strictly. And those involve what Dr. Mande said very importantly, shared crowded indoor spaces become a no-no. The use of masks, not so much to protect yourself, but to prevent you from infecting others. That's something people get it wrong. Masks are important when everybody wears them. If you're wearing a light surgical mask and everybody else is roaming around mask-free, the protection is actually not very high. And a few people trying to wear an N95 to protect themselves would be very suffocating over an entire day. So a general use of masks by people will continue to work, even if the infectivity rises. So that's point number one. A more severe disease. Now that's the one I worry about. So far, nothing has happened. The UK strain also does not seem to be causing any particular increase in severity, but there is some evidence that there might be strains out there that might lead to more severe disease, and we know it can happen. We know it can happen because MERS had a mortality of 50%. SARS had a mortality of closer to 10%. And the current low mortality is not because we have become that much better at treatment than we were in the time of MERS and SARS-CoV-2. It is simply because this virus is milder. And last, immune escape. There have been multiple experiments typically done in a way that you infect in a cultured cell and you incubate with some antibodies and you see the virus ability to escape. It tells you a little bit about immune escape, but true immunity inside the human body uses your T cells as well. So while immune escape from antibodies is well proven for some variants, as seen in this slide from Dr. Vinod Skaria's work, that again Tej spoke about at IGIB, you can see state by state, you can see the various mutations proven, well, proven is a very crude word here, thought to with some degree of experimental data to possibly lead to some degree of immune escape. And you can see many of those variants are already here. But overall, looking at the Indian scenario with infections continuously going down, probably a very large number of people, which in my estimate is more than 300 million, already infected and recovered, and probably innate resistance to infection. So you can have resistance to infection and resistance to reinfection, together accounting for probably something like 600 to 700 million people. That's what the supermodeling group of DST has been talking about. They don't talk about total number of infected people. They talk about people resistant to infections or reinfections. So far, these variants do not seem to have created much impact. But there are two states where infections have not gone down. One is Kerala, one is Punjab, and it will be worthwhile looking at this. But the total number of reinfected cases that have come to IGIB, and we've been one of the major sequencing sites, still is so small that I do not think that's a major problem clinically from a public health scenario. And the main reason for that, I believe, is not just inability to escape antibodies, but a wide repertoire of T cells created during an infection that provides longer lasting immunity. We have some work at IGIB showing that antibodies are stable at least until 6 months. It will be on medRxiv today or tomorrow. I want to focus now for the rest of my time I have on how should India be building capacity going forward. Tej talked about 5% sequencing. To do 5% sequencing at 20,000 positives a day takes you to about 1,000 sequences per day, which takes you to about 30,000 sequences per month. And to do that, you need to have a plan. And that plan can be what's called a mega lab, selected institutions with high capacity, and micro labs across the nation. What can a mega lab do? Many of you might think, India did only a few thousand sequences for the entire year 2020. What makes it think it can do that many? And the reason is technology has changed. On the Illumina NovaSeq 6000 platform, for which India has at least 5 labs, public, with the capacity, actually 6, and even more in the private sector. Now you can do something called COVIDSec, in which you can process 3,000 samples in 12 hours with full automation. To be conservative, I like to call this 1,500 samples in 24 hours because of a lot of upstream and downstream processes. It's actually a very simple process in which you run multiple samples with nucleotide barcodes together in a large single run. Ultimately, there's only a 30,000 kb genome, right? I mean, 30 kb genome. When we sequence humans on these machines, we are sequencing billions of bases. So from a data standpoint, we are ready. It just took this idea of barcoding, allowing multiple sequences to be processed in parallel and then automatically computationally resolved back into different samples and calls to be made for each sample. And this is a run that I'm happy to tell you was done for the first time in India. Of course, the technology is from a company, but the first proof of principle, proof of concept came from India, where 752 samples in duplicate were run on an S4 flow cell, which generates terabytes of data. And you can see that it functioned quite well at multiple CT value ranges. Now, if you want to really get high quality calling for every part of the virus, you need to only use samples with high viral loads. And when you pick your 5% samples, you can pick those with the highest viral loads, which is typically CT value at least lower than 25. And this type of sample collection, the program will follow the following ideas. A basic sequencing in which you're doing a fraction of the samples, and then there will be people where you want to sequence 100% of the samples. These would include immune international travelers, including those from UK. These would include people who are suspected to have a reinfection, people who are having particularly severe disease, and any large outbreak that is happening in the country. There you would sequence 100% of the samples. But like I said, there are only about 6 such machines in the country, in the public sector. And although that is sufficient to meet the requirements based on the numbers I told you about, it may not be practically doable because you would continuously be shipping samples. And if you wanted to know the answer fast, if you're putting somebody in hard quarantine and that person cannot get out of the hard quarantine until the results come back, for example, a traveler from UK, it is a strain upon the healthcare system and the patient themselves, and then they will not cooperate. So you need point of care sequencing ability, because otherwise it takes 2 to 3 days to ship a sample and then these runs will start. You can't keep doing a run every day. So practically you do one run every 3 to 4 days. So it takes a week before you get a sequence. So a micro lab needs to have a low infrastructure requirement. It needs to be affordable. It can have a low throughput, but it has to be reliable. The best current solution on this, and there is a very nice paper over there that you should look at if you're interested in this area by Maggi et al., of pore-based sequencing. You can have a biological pore or you can have a silicon pore. Every time a nucleotide goes through it, current changes. So current change can be seen in B. These change in current can be resolved into calling of bases and can lead to a sequence. The problem so far has been an error rate of approximately 5%, which makes it unsuitable for human use. But for strain identification and microbiological calling, it's a very nice technology. It's also very interesting that right at the time when India is thinking of starting these micro labs, a new paper has come in Nature Methods as of yesterday from Denmark and California, which shows how to reduce the error from 0.5%, from 5% to less than 0.5%. That is something that we need to look at, in which case it can become suitable for human use as well. There are also new versions coming for nanopores, which are purely silicon-based. And a silicon-based pore will be far easier and cheaper than a biological pore. Technology is still evolving. A company is called IMEC. Those of you interested should look at it. Very interesting area. The device is this small. The one on the one here has its own onboard computer. The one here does not have an onboard computer, but can be connected to one, including smartphones. The entire analysis can be done on the cloud. Very similar sample to sequence strategy compared to the Mega Lab. Again, you isolate the nucleic acid, you reverse transcribe it, you put primers, and you sequence it, and you can actually see it on your mobile phone kind of coming out like this. The advantage of a micro lab is that you can do it instantly, right there where the positives are being generated. If you had a mobile RT-PCR lab, for example, the way Spice Health has built. And number 2, you can keep adding samples. In the Mega Lab concept, you have to do the runs with a sufficient number of samples from the beginning. So it's a huge amount of coordination. But here you can keep adding samples as they come. There are certain flexibilities in pore-based sequencing. So I'm going to start I'm going to end here with the thoughts of what next. I hope I've proven to you there is a public health reason for which we should do it. On the WHO scales, India falls, and I'm going to show you that slide again because I really want to make a point of why India should take leadership. We have not only sufficient diagnostic capacity, but with falling tests, we have room to support. We have sufficient physical sequencing capacity, but with the micro labs, we have room to support others. And sequence data analysis. India is probably one of the most IT strong nations in the world, despite being what you would call an emerging economy. And I think these are areas where India needs to work together as a strong nation to support the entire Southeast Asia region and perhaps even parts of Southeast Asia. I'll end here. Thank you all very much.
Thank you, Dr. Anurag, for really elucidating, about the vaccines and the impact of these variants, whether should be worried or not. And you pointed out that as of now, it's not so much of a concern, and there is also the T-cell response, which by and large can take care of the protection for us against this. So thank you. Now I will move over to our next speaker, Ms. Avani Singh. She's a young entrepreneur. Welcome, Avani. Avani was born in Delhi, and then she's done a bachelor's and master's from Stanford University, and she is featured in the BuzzFeed's list of 25 women under 25. I think very inspiring that at such a young age, she's managed to do so many things that she's been featured in BuzzFeed, and she's also learned flying and was part of the Stanford Flying Club, and she has also a private pilot license. And before spearheading the Spice Health, where she's the CEO, she was working at McKinsey, and right now she's the CEO of Spice Health, which is a new venture. And I think Avani has worked with CSIR. They have taken two of the technologies, one from CCMB, the dry swab direct RT-PCR method, and the more recent one is the testing at the airports. So Avani will talk to us about how the industry is pitching in during this crisis, and how they're working with the government, and how this is going to make a difference, for example, in identifying the variants, etc. So over to you, Avani.
Thank you so much. Thank you so much, Dr. Geeta. Thank you, Dr. Mande, Dr. Anurag, Tej. I have immense respect for, a government research institute like yourselves, and I think that there is some incredible work that's being done. And I'm honored to be an industry partner and to help take these innovations to the market. Let me start. I think Dr. Anurag gave a very nice overview of the genomic sequencing project that we are doing at the airport, and made a very convincing case for the public health benefit of sequencing, not just for COVID, but also for infectious diseases, and eventually perhaps as a primary diagnostic tool as well. Let me begin by giving you all a little bit of an overview about Spice Health. And then I'll elaborate a little bit more on our pilot project that we launched just 2 days ago at the airport for genome sequencing through mobile laboratories. So Spice Health, we inaugurated about 3 months back when COVID cases were at a high and testing had reached its saturation levels, especially in Delhi. RT-PCR tests in particular were very, very difficult to find, and the price for these was also very high. We came up with an innovative model of a mobile laboratory, the advantages of which, of course, are that it is highly deployable, highly accessible, scalable, and cost-effective as well, because of which this model received a lot of government attention and support. And we were able to start conducting RT-PCR tests and take off a lot of the load from Delhi government to begin. We offered these tests at 499 rupees as opposed to the existing rate of 2,400 rupees in Delhi, and are now conducting about 15 to 20,000 RT-PCR tests a day, and also quickly scaling up to states across the country. As I mentioned, I have immense respect for research organizations, and I truly believe that working together helps bring this research, in a commercially viable model to the general public, and really we can really make a difference in the country through such partnerships. We did adopt the dry swab technology developed by CCMB as well, which of course helped us save a lot of time and price in our RT-PCR tests too. So that was again an incredible innovation that we are proud to have adopted in our laboratories. And now we've embarked on a genome sequencing project, which we are very excited about. We have launched genome sequencing in our mobile laboratory at IGI Airport in Delhi. This is a regular 40-foot container, which of course has potential to be reduced in size a lot and hence be made far more scalable than it already is. This is a pilot project. We'll be assessing the viability of this project on 3 major factors. The first is, of course, timeline. As Dr. Anurag mentioned, and so did Tej, challenges lie currently in the current structure when samples come in from international destinations and they are tested positive for coronavirus at the airport itself. They are then transported to government laboratories across the country. That transport of the entire sample firstly takes time. Extraction of RNA then takes further time at the labs. And since you cannot run all the samples together if there aren't enough in a particular day, by the time results for those samples come in, it could be a week to 10 days of time. This means that the people coming into the country who are in hard quarantine can get frustrated, not knowing whether they have another strain of the coronavirus present in them. And if they don't, they can be moved to a soft quarantine facility. So this adds a lot of stress and burden, not only on the country and on our infrastructure, but also on individuals itself, just as Dr. Anurag mentioned before me. On the other hand, there are, of course, folks who are put in soft quarantine who may have a strain of the coronavirus, that is the UK strain or the South Africa strain or any other strain that Tej had covered that we may not be aware of yet. And they would need to be then instantly moved to a hard quarantine facility to ensure that they're not transmitting this virus further. That's another burden, not knowing for 7 to 10 days and someone infected with such a virus being in a soft quarantine facility. So timeline, of course, is one factor on which we are going to be judging this project. Another is accuracy. We'll be seeing if the samples which are tested in this nanopore technology at the mobile laboratory at an airport holds up to the standards of an Illumina or any other technology that's being used at the mega labs across the country. And lastly, we will, of course, be assessing the cost benefits. We believe that the cost benefits could be immense, especially since we are not transporting the samples anymore to faraway facilities, and the testing is happening at the location of the RT-PCR test itself. This will give the health ministry enough of a sense to determine whether it's viable to set up more such laboratories, not only at airports, but also at other remote destinations where testing is happening currently. So this is the pilot project that we have launched just 2 days ago. It was inaugurated by Dr. V.K. Paul, which we were very proud that he came for the inauguration. And we look forward to seeing the benefits of this 30-day pilot program on sequencing in the country. So that, of course, is the latest thing that we have been working on. And like I mentioned, personally, I'm not from a medical background. I'm a new entrepreneur. And I'm very, very proud to learn every day from talks like this and from Dr. Anurag, Dr. Ray, Dr. Mande, and all the incredible scientists in our country. And I feel very, very honored. And I think I'll open it up to questions now and keep it brief. But thank you so much for having me today.
Thank you, Avani, for really bringing in the component of how industry is really trying to help, in bringing down the cost and increasing the accessibility, because that's, I think, a key important issue when you're dealing with the pandemic. I hope it's successful and replicated in other places as well, because I think although in India we've started the vaccination, international travel, as long as there is international travel and we are in a connected world, I think we have to be on the lookout and have surveillance going on in the country. I think that's a very important aspect, and low-cost solutions like this, I think, will go a long way in actually keeping the country safe from the future variants. I hope, I wish you success in your future endeavors as well. And So there are a few questions, I think. So I will tell the question and so how viral clade differs from a lineage. So Tej, perhaps you could answer this.
You could, I mean, clade and lineage essentially mean the same thing. Actually, clade is a group of viruses which share a common ancestor, or also the trajectory is the lineage. So yeah, both are the same thing.
Yeah, that's good. Yeah. Just 1 second. Somebody is asking, how do we ask questions on Twitter? So I was just responding to that. Yeah. The second question is, Anurag, perhaps you could answer this. How is the immunogenicity or pathogenesis severity for different variants or lineages?
As of now, there is no clear evidence that there is any particular lineage with a very different severity. In the beginning in China, when A and B strains came out, there was some indication there might be a bit of a difference, but at this point, with the pretty much across the world, the experience has been largely similar within nations, irrespective of clades. Now, I'll qualify the statement by saying the experience of severity has been extremely different across nations, but not attributable to the viral lineage. And why it is different between nations probably has less to do with the virus and more to do with the humans and the way their ages, their way of living, and their background exposures.
Okay, Anurag, there's a question from Priyanka Pulla here from Twitter. Can we learn more about the N440K? Is it associated with immune escape? How was this established?
Okay, so N440K is one of the variants that we are already starting to see. The immune escape, like I was explaining, are mostly coming from in vitro experiments where you take either you predict binding sites of antibodies and you look for changes in those domains on the spike protein, or you infect a human cell in vitro, you add antibodies which are known to affect a particular area, and you look for the virus escaping and being able to infect other cells. Now, both of these are important, but they do not contain the T cell component of the immunity. I would only take immune escape seriously when I start seeing actual increase in cases of reinfections. Now, there are two caveats to this. In a country that has not tested enough to know its infections, reinfections will not be easily found. But there are enough number of countries in this world which have done plenty of testing, and India, especially with the advent of the growth of testing, is joining them. So I would say that I would look for clinical evidences of rising reinfections before I take immune escape very seriously.
Okay. Yeah, so I think the T cell component is an important aspect, like you mentioned in your talk as well, because and these are in vitro studies. So actually, unless you see in the humans, you were not sure of the whether it will.
I will add one thing. So T cell immune measurements is something that is actually not very difficult. What you do is you take frozen PBMCs, you transport them from patients, you expose them to the virus, and you look for proliferation and response. Now, there is some very nice data that at least in some countries, about 30% of the population, when cells are collected even before SARS-CoV-2 was there, show responsiveness to the SARS-CoV-2 viral antigens, which basically tells you that in every population, there will be a background of people who are resistant to the first infection. And this number could be anywhere from 10% to 50%. And one of the reasons people are thinking that some of the tropical countries like India have done better is because a chunk of the population might be innately resistant because of high amount of viral exposures, particularly in area like slums. Now, these experiments have not yet been done formally in India, but CSIR is gearing up to do these as the vaccination starts, because when we collect the baseline samples of people, we will get these numbers and be able to compare them to the globe. And then when we follow people who have been vaccinated, we will also be able to understand how these are changing. And until these are added to the antibody tests, I think we are deficient in our knowledge.
Yeah, so this is part of the surveillance, post-vaccination surveillance study, which I think CSIR is planning to embark on.
Absolutely.
Right. Yeah. So there's another question. Could the mutation in spike proteins as seen lead to the failure of existing vaccines?
Okay, I'll take this. Could it? Yes. There are plenty of examples in history where mutations in viruses led them to escape previous resistance to other viruses. In fact, I would argue that SARS-CoV-2 itself is an example of a virus that broke through all the previous immunity to coronaviruses. Does it appear likely from isolated mutations here and there? Probably not. But a more complex reassortment, rearrangement coming especially between host, human to animal, jumping back and forth could do it. So which is why zoonotic surveillance will also remain important.
Yeah, I think most questions are to you, Anurag. In early days, vaccines were developed, and basically the question is it took more time to develop vaccines. But now vaccines are developed in a very short time. Is it correct use public in the sense that they are concerned about the safety aspect? Okay. I think.
Let's basically think about this for a second. How many ways you make vaccines? Ultimately, a vaccine is always about presenting an immunogenic, which means raises immunity, part of a pathogen, to the human immune system in a way the human immune system reacts, matures, and develops memory cells to react to it if faced with a real infection in the future. Now, the original versions used weak or killed viruses along with something called adjuvant. This is very proven technology, very well established. The Indian rotavirus vaccine, for example, is one of those. Classical polio, smallpox, everything is. Basically, all old vaccines are this technology, killed or weak viruses. Now, when you do this, what are the limitations? The limitation is you are culturing a virus, you're growing it. It is not exactly the same thing as a virus infections inside the body, but it is similar enough. So you expect to get, 50, 60, 70% type of immunity. That has been the routine over the years. If you're lucky, like with huge viruses like smallpox, you got great immunity, but that does not typically happen for the type of viruses we are talking about. So low but useful immunity for public health purposes is expected from such vaccines. Now, in today's scenario, the equivalent vaccine in this would be the Sinovac from China and Covaxin of India. And immune response studies have been done. Of course, the exact percentage efficacy remains to be shown, which is why the government approved it in a clinical trial mode, which basically is another way of saying we will figure out the efficacy, but we believe it is likely based on everything that we know about the science. The second category of vaccines do not use this virus, they use a different virus. And this virus has been fed with the instructions to make the proteins of the virus to which you want the immunity. And the adenoviral constructs of the Russian vaccine, the Oxford vaccine, of course, being the Serum Institute vaccine also, are of this category. So using a different virus, which is innocuous to get into the body, but that virus makes the proteins and immunogenic regions of this virus, and you develop immunity. Again, very well proven, very safe, medium level immunity. So, the 70% claims of Oxford AstraZeneca sound quite likely. And importantly, both the old vaccine that I talked about and this vaccine, the overall efficacy might be 70, but the real concern is about preventing severe cases, right? If all of us only got the flu and we recovered, why would we care? And in both of these types of vaccination, it appears that the reduction in severity of infection might actually be greater than the prevention of infection. So that's something to keep in your mind. The last one is the mRNA class of vaccines. It is a brilliant strategy, beautiful science. You say, why do we even need a virus? Let's just take the instructions, the code, the mRNA, inject them directly into the humans. It is technically much more difficult than it sounds. Let's make the human cells directly make the protein. The muscle cells are ideal for this. And that protein will get exposed to the human immune system over a long period, and you will get very high immune response. That is exactly what we are seeing. Safety appears to be very good. But let me say something from a clinical standpoint. Safety is greatest when a large number of people have taken it and the experience and the data is over a very long period of time. The mRNA vaccines we are seeing today are the first of their kind in history. The reason we got there so fast is because they are mRNA vaccines. You can start planning them and designing them as soon as you get the virus sequence, which again comes back to why sequencing was important. But long-term consequences in terms of immune stimulation need to be seen. I don't think there's going to be much. I mean, after all, we all do get viral infections, we recover. All the other concerns people have regarding, integration into our DNA, etc., is not going to happen. It's mRNA, for God's sake. It just goes away after that. But yes, that's roughly where we are.
Okay. Thank you. I think you really took us through the 3 different approaches of vaccination, and each has its own merits and, I think, challenges. But that's the way research is, I guess. So, there is no easy solution here. So, Tej, maybe this question you can take. How can we block the expression of ACE2 receptors?
It's an important receptor, and I don't think we should think about blocking the ACE2 receptor. So I think that's a good. There are ways to do that, but that will probably kill the cells. So let's not think of this.
Okay. Yeah. And okay, how does again? I think you answered this question already. How does the current vaccine that is being provided affect the new variant of SARS?
They probably mean the UK variant.
Probably,
and like I said, both vaccines, one takes the whole inactivated virus, one takes a very large part of the spike protein. Multiple epitopes are covered beyond the ones mutated by this UK strain. So I do not expect there to be any problem whatsoever.
Okay. So somebody is asking, I don't know, I don't get the question perhaps. As we talk about SARS-CoV-2 variants, how is it different from bacteria? Probably they mean, the mutations in bacteria and, bacterial mutations also happen. So how is it different perhaps? That's what I would think.
Well, from the point of view of today's talk, they're similar. So you could have mutant versions of tuberculosis with greater lethality, greater spread, immune escape, all those are possible. For any pathogen, pretty much the ideas are the same. With viruses, the number of proteins is very few because they are basically obligate intracellular organisms that replicate only inside cells. And the importance of T-cell immunity is greater perhaps in viruses than it is for other pathogens, but that's a loose statement. It depends on which pathogen.
Okay, I think, yeah. So I think we have no more questions, and it was a great discussion, and I hope the audience also enjoyed it as much as I did. And I think the future is, the way I look at it is all these innovations, although they are meant for SARS-CoV-2, I think they are going to play a big role in India's healthcare system. Because all of these can be used for other diseases as well. And I think, so that's the learnings from the SARS-CoV-2 is that I think whatever innovations we are doing here and the efforts put by the government, the industry and individuals, I think is really going to help boost India's healthcare system in future. I think this point of care diagnostics and this mobile laboratories, these are really, really going to transform India's healthcare. And with that hope and the vaccination beginning today, I would like to end the webinar and thank you all for participating here. I really enjoyed it and thank you once again.
Thank you. Bye.
Thank you.